aspect or the drought tolerance, how it helps in drought tolerance. For that, we check the root architecture analysis of this gene. There also we use different approaches. The first one was rhizopenal method for root architecture analysis, where the seeds was germinated on a germination paper, which one side of which is immersed into the Yoshida culture, and the solution is taken up to the germinating seed with the capillary action. And this germination paper was sandwiched between two plexiglasses, grown for 21 days with the constant imaging at the constant interval. And then we scan the roots to look at the primary roots, nodal root, primary lateral, and secondary lateral roots. The results of which, after scanning and analyzing, we see that there are more main root length, lateral root length, and then number of lateral roots are more in the knockout compared to the wild type. Also, the lateral root diameter is increased. The second one was soil in pot method, where we keep the control at 100% soil moisture capacity, and the drought, we stop watering the drought treatment after 21 days and check the soil moisture to come up to like 30 to 35. Then we carefully wash the roots from these two, scan them using the WinRhizo, and then the analysis shows that there is increased total root length, root volume, and average root diameter in the knockout again compared to the wild type. The third approach was mylar tube, where we use the mylar tubes, which is easy to handle, and where we fill the mylar tubes with same quantity of soil, and then keep the control soil moisture at 100% capacity, and then stressed at 70% field capacity until 21 days. And then we stop watering the stress one. We sample them after 30 days. So we remove basically the core. This is the core, the whole root basically. And then we wash them carefully, scan, and then analyze it. Here also we see the total root length, lateral root length, and root diameter increased in the knockout compared to the wild type. But in the root diameter is mostly increased. Looking at the root hair and length and root hair density. Okay, before going to that, I'll say that all these three experiments shows that clearly that the knockout, main root, lateral root, and number of lateral roots, we see the increase. Okay, looking at the root hair density and the root hair length, we see both in control and the stressed condition, the knockout shows increased density and length of the root hair, which is very important in the drought tolerance and nutrient uptake. We also looked at the sections of the roots, and what we see that the knockout shows more parenchyma under well water condition. However, under drought condition, it shows increased diameter of xylem vessels and reduced parenchyma. And literature says both of these observations are important to contribute to the drought tolerance for the plants. Okay, so coming to the conclusion or summary of this whole presentation, that this is the first report of guanine deaminase gene in plant. Our phylogeny, protein phylogeny analysis suggests that the unstable guanine deaminase was selected throughout the course of evolution by farmers and breeders for root rate or grain yield under drought, and hence we call it a backbencher gene. The guanine deaminase has putative role in epigenetics of drought tolerance through DNA methylation. There are several reports. There are like, I wouldn't say report, but we have done extensive analysis of, for this we calculated the SH content and guanine content and xanthine content in wild type and knockout. I haven't shown that data here, but once the paper is out, you can see that in detail. And then null allele of this guanine deaminase is responsible for beneficial root architecture under drought stress. Before finishing up, I would like to acknowledge some people. First and most important is Dr. Ajay Kohli, who has been my PhD supervisor during my time in IRRI, Philippines. Caroline from Sinica, these are our collaborators. Kenneth Olsen from USA, Maria from Spain, and Biswajit from IISc India. Also my colleagues and mentors, Toshi, Amrit, and others who have helped and kept the environment in the lab friendly. And not but least, I would also like to thank Government of India for providing the scholarship to study this research at International Rice Research Institute. Okay, thank you, and we'll be happy to take few questions.
Okay. So you can see in the chat. The questions are being pasted in the Zoom chat.
Okay, the first question is there an ortholog of OSGDA1 in barley? How well? The first half of the question, if it is present in the barley, I'm sure it is because most of these genes are like, we call it amido hydrolase, but if you look at detail into the what is the amido hydrolase, there are also synonyms called nucleobase nucleotide hydrolases. I'm sure this is orthologs are present in barley, but I haven't checked it by myself. You may check it. You can put nucleobase nucleoside hydrolases. Second half is how well conserved is this gene in plant species. It is conserved, that's what, it is conserved, but haven't been characterized into the plant. There are some reports in Arabidopsis, the guanosine deaminase, guanosine hydrolase has been characterized, but in Arabidopsis, but in the crop species, we haven't come across. Specifically, as I said, this is the first report of guanine deaminase. So nobody characterized guanine deaminase in any plants till now. Okay. Okay, there is one question called, is there any other rice varieties where this particular gene has been assessed and studied? Is it only for indica only, or we see it in japonica? Actually, as I said, one of the parent is indica, one is japonica. Also the knockout line we checked, we did the metabolite analysis for that also. The knockout was from the T67, that is Thainung 67, which is a japonica. So, and also the phylogeny analysis, if you can see, the phylogeny analysis been done throughout the panel, which include the japonica, indica, temperate japonica, tropical, all the clades. That's where I call all the rice accessions and land races and all of them. There are two questions. Did you also record the root angle, root architecture? Is there any relation to root angle in drought tolerance? Yes, it is. We did the shoal analysis for looking at, and then what shoal analysis shows actually, it shows the, like a graph, you know, like a peak, we call it the hyperbola. So when we see the increase in the peak, that means there are more chances of the branching or making the branches into the root. And that's when we can calculate the angle. I haven't shown that study in here. Next is there any study of this drought tolerant gene in other cereals? To our knowledge, no. This particular gene have not been studied anywhere else. As I said, this is the first report of this gene. Okay, there is another question. Do you think that the root density of the rice variety affect the drought tolerance in rice? Well, there are different hypotheses or different studies, actually, not hypotheses, different researches have been done on the density of the root or the length of the root, what can contribute to the drought tolerance. I would say it depends on the soil condition, it depends on the environment, it depends on the variety you are using. Some varieties tend to not go very deep, they tend to be shallow, like they just, so basically what they do is they spread out their root on the upper surface of the soil, like making the branches, increasing the root hair length. But some varieties go deep, like they dig deep to withdraw the water from the deeper surfaces of the ground or the soil. So it really depends where we are looking at. If you are looking at, very loamy soil, that depends. If you are looking at stony soil, that depends on the soil and the environment. But yes, density affects for sure. There is one interesting question. Can you explain the relation of ground root produced in Yashoda solution with drought stress? We haven't looked at it, but that's interesting. I could dig on that. We haven't seen. Um, okay.
It was wonderful. Today's webinar, people have appreciated it so much. You can see from the Zoom, YouTube chat.
Okay.
Okay,
Thanks.
the show has to say something. He's back.
Yes, I'm back. It's nice, very comprehensive and concrete work, and I love your some instrumentation. Like it's I wondering how we isolate the roots of a rice, right? Because when we go for the Yoshida, it's okay, but how we could isolate the rice, and we remember Jacinta, we when we the IRRI that. We go for that type of training also, that how you bore the tubes, the metallic tube on the rice and pull the rice and just
So much work.
eject the soil. So it's I am sure this type of things are also very interesting to learn. It's not a data, it's just a technique, but sometimes it's answered to design methods or these things, this type of experiment. But sometimes we just not doing that because we don't know the technique.
Exactly.
Okay, so it's a very good perspective, and rice root is always fascinating. It's always fascinating because when we did in the Yoshida, that the vigorous of vegetative growth is so high, and we love to photograph, and these things of the root is so transparent because we got so many related queries about the roots. Okay. uh, and we have the queries when we don't see it. We see the leaf, we see the fruits and stem, everything, but yes, roots is very interesting, and I'm glad that you are working on that. And for an agronomist perspective, it is also very important. I am sure it's always helpful. So we have a few minutes. I want to stay or 5 to 10 minutes to that. So let's I ask you a few related questions, but not the talk or thing, because it's visible in the YouTube chat box, and I think many of the listeners, they know you, or they know your work, they are appreciating you in that chat. So another is I'm asking you that as you now agronomics, that the distances between two plants, like that when we do in practicals, basically we did in a pot because our plants are transgenic, so we not go for the direct seeded or in the plant in the soil.
Field. Mhm.
But do you think that these 10 centimeter gaps, which is generally everyone as a thumb rule that 10 centimeter gaps between the two rice. Uh, have you think that when you follow these rules about the distancing between the crops, we think that it is for the leaf and the panicles. Do you think it also affect on the roots? If you create the distance, then how it affects the root development?
Well, before the root gaps, actually, yes. The distance between two plants we keep at least like 15 centimeters. Um, we look at the leaf and the panicles, which is above, we call it above ground plant parts. But the below ground plant parts are interconnected. You must have seen the studies where the one plant root has grown underground, which releases some hormones or gives signals to the other plants. Okay, so it happens mostly into the symbiotic root systems, where one plant gives the signal and then parasitic or the symbiotic plants get um, involved into that. This, I think, similar reactions happen in terms of the roots into the crop fields, because when the drought comes or the flood comes in the farmer's field, it doesn't come like suddenly into the whole field at a time, right? It comes into the patches, like depending on the soil density and then the water, relative water content into the soil. So these signals comes from one part of the field to the other. But that's the soil root connection. But when we talk about sampling these roots for our benefit or for our experiment from the field condition, as you mentioned, that metal pipe into the field. So basically what we do is, it's impossible to get the single plant root into the field condition. First thing is distance is less. Second thing is we don't know. The roots are only going deeper or spreading out. If it is spreading out, for sure there is an interconnection of the roots in the below ground part. So it's impossible to take the single plant root. So what we do is we call it a coring of soil. So we core in between the four plants or two plants. So what we get into our core is a mixture of roots from two plants or four plants, and then we sample, which is also good because we get the random sample size, so we can cover more. So there is a more randomization along with the growing the plants, and our data we can believe or stick to our data more reliably than getting the single plant root.
Okay.
Okay. And another interesting query for my end, that we know that if the drought is high, then the loot trend is increasing. Okay, to try to capillary actions and take the water from the more deeper soil. If it is drought is so affected on the roots. So do you think there is a limit, or obviously a plant can sustain a certain limit of the water purity? If it is so high, then plant can sustain and everything is stopped by the development, and ultimately plant will die. But uh, I want to know that whether is there any rapid developmental change after the certain time of drought. Means if it is 5 hours, 10 hours, or 3 days, because sometime we watered our plant after 1 day interval, or we try to keep the water level 1 centimeter or 2 centimeter. So I want to know for my own purposes that if we forget or we don't give the water, then when it starts to developmental changes due to the drought.
Sure. Actually, this is also a good question for the listeners also, actually. What are the changes? So your question, Shiva, is what are the changes you can see? So basically the physiological changes. If you look at the molecular changes, it happens within minutes. Okay.
Yes.
But the physiological changes which we can see is not only below ground, but it is also the upper ground. There are several mechanisms to combat to the drought stress or heat stress. Below ground, we know the root length and deeper roots and root branching and all those things to dig out more water. But I would also say above ground, there are changes. It depends on the variety. Some accessions have this capacity to switch off or close and open their stomata, okay, to reduce the transpiration. So when they have water, to save that water, they can close and open the stomata according to their convenience. Also, there is a leaf rolling. So, the leaf is like wide open, but when the direct sunlight hits the leaf, it also affects the stomata, and then it takes away the water, in terms of transpiration. But then what the leaf does, this happens in all the grassy leaves. What the leaf does, it closes itself. So we call it leaf rolling. So basically it rolls inside, not outside. It rolls inside. So when it rolls inside, it reduces the surface of the leaf, hence reduce the translational loss of the water or the moisture. This is one thing from the above ground. Also, some plants stop the growth. If it is hitting the drought or the heat stress, which is more than the bearable capacity of the plant, it will stop growing. So in majority of the cases, you will see when in my image also, you might have noticed that when I show you the control and the stress plants is most of the time is the height is short compared to the control. So that is one mechanism. It stops growing. That means it uses that energy, that metabolic energy, not to grow above, but to grow below in the roots. I have seen a rice variety which is called Moraberican when I was working during my masters in Bangalore. The roots of Moraberican could grow in drought condition up to 1.5 meters. Okay, that is an average or more than average of a maize root length. Okay, you can see the maize root, how big it is and how big it could go down. But for like rice, the average is like 60 centimeter, 30 centimeter, 45 centimeter. I have seen the maximum average like 45, 50 to 55. But this Moraberican one could go maximum 1.5. That is like 90 centimeter. Even more actually, there were some Moraberican, there was one called AM6, which the maximum I have seen 112 centimeter. That deep can go.
The drought tolerant varieties, what you're talking about?
Sorry?
These are the drought tolerant varieties you're talking about?
Yes, these are the drought tolerant varieties I'm talking about. This material was selected in the Harvest Plus project, the rice Harvest Plus project, to enhance the zinc and iron into the rice in India. So we have the Ravindra Babu from DRR. They had selected these 180 lines throughout the India from north to south. And I was testing them for drought. Of course, I was checking the grain, iron and zinc content, but as our lab was mostly working on drought, I was testing them in the field for drought. And that's when I measured this root volume, root length, root diameter, number of roots. And this is the observation that I have seen the root length up to going beyond 100 centimeters.
Okay, thank you. Another questions I got from the YouTube chat. It's totally depend on the lab, but sometimes it help to listener. They Pawan Nakpal, wanted to know that what variety should be preferred more for drought tolerance analysis. So it's also depend continent to continent. All varieties not available. Vietnam variety is not available in India or not very popular in India. But if you give 2-3 names, so I think it will help to listeners, and those are the try to start PhD and that they can use as a sample.
Yeah. If you look at the drought tolerant and drought resistant, or drought adaptation, I must say, these are again the same. These are the different approaches of the plants in the different environments, depending on the soil condition, environmental condition, And although some varieties tend to stop growing, as I said, some varieties tend to keep growing, but then close their stomata or increase the root length or get more nutrient to get more energy so they can survive. Actually, this I haven't seen this in drought tolerance, but in flooding, this usually happens that plants store their energy, like these sub 1 varieties. They save that energy when the flood comes, and it's like waterlogged, completely plant is submerged into the water. They stop growing. They don't stop growing because they want to save that energy, but what they do is they stop growing. They wait until the water resides and they get the fresh sunlight so they can grow again, and the growth is very fast after the water resides. In terms of drought tolerance and adaptation are two different approaches of different plants, like Swarna. So in my panel, when I was checking these 180 lines, there was this Morobericon, which grows faster. It adapts to the drought, but there was this Swarna actually. It tolerates actually, or we can say resist. It never grow actually. So it's the total height, the above ground height of the Swarna is like 30 centimeter. And in control, whatever it's control or drought, it kept that variety kept its greeniness throughout the season. It didn't wilt, it did not dry. It was green all the time in drought and control. And it did not overgrow, it did not stop growing. And the result was like, of course, it did not show any great promising high or low results. It was stable. So basically it resisted the drought for some time and kept its maintained its growth.
Okay. Another question is, I'm sure the answer will be yes, because they ask, it is depend on the soil quality. Means the question is the crop tolerance depends on the type of soil also. So
Yes.
obviously,
Actually,
yes.
yes. To give you a very, very quick and simple and reliable example, if you go to the rice field, whether it's a research field or farmer's field, you might have noticed that you have a field of like 2 acres, okay? But among the 2 acres, you see a patch of like less than a half acre or like 10 square foot patch in the field where the plants are showing the yellow leaves, okay, turning yellow or turning reddish brown, okay. And you must have wondered why this is so. With within the plot, within one field, there is a patch turning yellow or brown or reddish. That may be because of the iron toxicity, because that patch, when you apply the manures and fertilizer, that patch somehow might have received extra iron fertilizer, or the soil already has more iron in that patch. So this is a very uh, relatable example to that. So this is how the soil affects.
Yes. Another questions is maybe related to for the question is that what is the reason of leaf rolling? Okay, so is it for the drought? So it is a question that what
You already
leaf
said,
rolling is the reason of the drought.
Yeah. I will answer your question, sure. But the previous question, I want to add in the previous question, answer that the soil type question. Um, yes, what I wanted to say is the soil compactness, because this is one I observed when I was doing the experiments in pots and the mylar tubes. The more compact the soil is, more hard it is for the root to penetrate into the soil. Okay, this is common, yeah. In our life also, when we dig the soil, it's hard when it's more compact. But when it's loose soil, it's more easy for the root to penetrate. So sometimes plants also react to this compactness of the soil. If it is more compact, it will try and put more energy below ground. And if it is loamy, it's easy for them to get the water. Okay. Coming to the current question. What was that show? Leaf rolling, right?
leaf rolling.
Okay.
Yes.
How the leaf rolling, is it for the drought? Yes, it sits for the route and also for the heat. We have people have also observed the same phenotype for the heat tolerance or heat resistance, because it's all the above ground part. Whenever the heat increases beyond its tolerance, it will roll it to reduce that surface, because sometimes plants need sunlight, the heat and the drought. Okay, because their main motto is to save the water in both the
Yes.
cases.
Yes. My last questions, and before going to that question, Shoma, can you? Actually, I'm log out from the YouTube. Can you
I will, yes,
write in writing that those who are not getting the link because they are
I will
not
also say it.
registered, you ask them to email us with the password. If password is not mentioning in the email, we're not giving the feedback link. Okay.
Yes.
Okay. Danny, my last question is, IRRI's 3K genome sequencing data, uh, is it helpful to your research as it is?
Yes. Shows network. Okay.
I repeat, actually, my internet connections is a little bit down. My last question is that, first of all, very thank you for joining. If it is not working the internet, then uh, it's very. I want to very congratulate to you that it's a nice presentation. If you presented your work very nicely. My last question is. Because you are from IRRI, and we also use this platform for our rice research. Uh, is it 3K rice genome sequencing data is helpful to your research?
Yes, yes, of course it is. And it is helpful not only for me, for all the people who is working in the rice field.
Yes.
Because the most important thing about this data set is it is free. No, you don't have to pay anything to use that sequence data. They have done the resequencing of, after the 3000 sequencing, they have also done the resequencing of 7000 lines. Also, there is the HRDA, which called Hydra. We call it Hydra data from Susan McCoy's lab, which is also incorporated into the 3K website. So basically, you get access to a lot of more than 3,000 rice varieties, SNPs, basically, non-synonymous and synonymous SNPs. And also, not only the SNPs or the genomic sequence, they have related that data, SNPs data, with the phenotype. So the physiological data, like for the drought and leaf, length, flowers, grain yield, then the anthocyanin, color tissues, the height, all these traits, they have correlated this phenotypic data with the sequence. So basically, if I don't know if you have used it, but when you go to the website, you can select your gene, okay? Just put the coordinates of your gene and then select the panel, which panel you want to see, 3K panel, Hydra panel, or whatever. Or you can also select the varieties which you specifically want to look at. And put the coordinates, select the panel, and hit the phenotype. The drop-down menu will also show all these phenotypes. I think there are more than 100 phenotypes data there. You can select whichever phenotypes you want to see. And then, search. After that, you will get an Excel sheet with, your coordinates, SNPs, synonymous and non-synonymous SNPs in all those lines, and how it relates to the phenotype. We have done the similar thing for, recently we have published one paper from Sinica on the HD1 gene. We have done this analysis for the plant height in that paper from the 3K data set. You can access that paper as well. Yeah.
Okay. Uh, Thank you. Thank you for joining with us. And it's